Very brief lang na siya because we are going to include application to genetics maybe if we still have time sa genetic engineering last naman na siya nga part. Okay? So second kani siya chromosome basis of heredity. Usually akong gigamit ani is si Campbell. Okay, si Campbell ang ginagamit namin. And then third topic kani siya, this is basic elementary genetics ni siya kaning segregation and integration ka Mendel, and then extensions to Mendel included na ni siya, law of inheritance and law of independent assortment and law of segregation, then extensions to Mendel. So this is actually quite a long topic or long mga 2 or 3 chapters niya sa book ni Griffith. So ako ang gamiton aning kaning third kay sa book ni Griffith kay very well discussed this book ni Griffith. Although this is also well discussed in Hartel and Hartwell. Pero mas okay para nako ang ka Griffith o ka Hartwell. Sa linkage and recombination, this is well discussed by Daniel Hartel. So maybe we can use Daniel Hartel as basis for this topic. So just like how we did in, or just like how we usually do in our courses, we tend to jump from one book to another depende sa topic. Okay? So kabalo na mo ana. Chemical basis of heredity na apo ni siya sa Hartwell, Leyland Hartwell. Gene function, proteins, and enzymes is well discussed in Campbell as well as in Hartwell. Na naman may copy ni Campbell, no? And you know how to download books from livegen.is na. So if you wish to download the recent version or recent edition of the book, feel free to do so. And then genes. Actually, there is a separate book for this genes. It's titled Lewis Genes. Wala siya na ako na-upload sa Google Classroom. Pero if kuan, ako to siya niyang i-upload. Pero kaning genes in development, napo ni siya sa ka-Hartwell, sa epigenetics. Okay. So, this is more on gene expression already. Okay. And then mutation, this is also discussed by Anthony Griffith. Sa bali na pud ani ini mabot as mutation. Bali na pud aning Griffith nga book. Okay. Kay mas kumpleto si Griffith kaysa sa uban nga books as I have observed. But makagamit raman gyapon mo sa ubang book. Delayed chromosomal and extra chromosomal inheritance na ani siya sa Hartwell. Okay, later na ni siya nga chapter 13 wa ko nasayop. Nasa dito na unahan. Okay. And PopGen, kani siya is discussed in evolution man ni siya. Kaning PopGen, Hardy-Weinberg law. Ani siya dito sa kuan. If makaabot ta ani kay 11 naman ni siya. Hopefully makaabot ta ani. So, ang sa genetics, sa may atong paagi ani magkuan man ta naman ni solving. So, open cam ta ani guys. Okay ra? Naara man may connection, di ba? Syempre na jud may connection. Requirement man na. Di po pwedeng mag-record ako lecture videos online with ani. So, wala mo'y choice online. So, ang atong solving natay drill. Kung sa face-to-face pa ni, pasulaton tatamo sa board. Nya kay dili man. So, siguro, prepare lang mo og kuan, paper and pen, and then open cam lang ta. Dayon, paman inyuha lang, ay di po mo ka-share screen sa kay na mas inyuhang paper langan. Maka-open, maka-share screen ba mo using camera or dili? Naka-try mo ana? Kung
Try daw
mas,
nun, ma'am.
Wala, ma'am.
I-try daw.
Tryan daw.
ako'y nasuguan.
Kami may matry, ma'am.
Kaysa, kana, okay, makita ra di ay guys. Makita ra no?
Hmm.
O, sige, ato
Pero
na lang
sa
tong
phone
sign.
lagay, ma'am.
Oh. Oh, pwede sa laptop. Klaro naman po dago inyong ang inyong camera sa laptop. Pwede ra. Patungod ra
2
sa
MP.
inyo. And then you can show your unsa ni inyong tawag ani. You can show your answers and at the same time explain ana lang ta para nagya po drill and practice. Okay. So kana siya naana siya sa gene segregation and integration nga part. Napo na siya sa PubGen. Napoy solving ang linkage and recombination like kuan na mangud kaayo mo sige tag solving di man pud tabi is bio major in mathematics no. So kuan lang siguro will focus mo sa kuan lang sa concept sa linkage and recombination. So the usual. So after explanation ako sa kuan topic, for example, chromosomes based severity, I will give you kuan article presentation dayon. Schedule ra ta. Okay? Kaning PubGen mag-discuss ta ani murag mag-discuss man pud mo ani sa evolution kay discuss man is um yeah, I will still discuss this despite being discussed in evolution. Ang laboratory, maybe I can give you lab activities, but
Oo.
in genetics manggod, ang lab activities does not require sophisticated equipment. Okay? So, maybe we can have lab activities for genetics, answer that, and then submit it via Google Classroom. I'll just post the requirements in Google Classroom. As for your requirements, semestral paper, gihapon ta, review paper for lecture, and then just focus on review paper na lang siguro. Kay wala man gud bisan pag maghatag ko ninyo og research paper man gud wala tay equipment available in our institution so it's quite useless. Murag mag-imagine ra ta ani og PCR, diba? Imagine tag PCR and then mag-imagine po tag chemicals, imagine ra tanan. So, just pour your energy na lang sa review paper siguro. The same gihapon, I will be asking you to submit your topics subject for approval before you can continue, ha? Sayo-sayo lang po ihatag ang inyong mga topics.
Pwede
You
mo
can,
hatag na lang kag deadline, ma'am.
um,
Last.
deadline, it's up to you. If you wish to start on the last month, then start on the last joke. Bitaw, ano man ron?
Para sa
April
proposal,
8.
proposal nga kuan,
Next
ma'am.
week. Oh, no. You still have to read man, no? So, I'll give you
Yeah.
1 week siguro. The deadline for your review paper topic will be next week, Friday. Okay?
By pair, ma'am.
No, individual.
Individual.
Yes. So if we still have time, I will be requiring everyone, if we still have time,
Ay, pila ka book review paper ang mo.
usa, tagsa mo.
Ay, di ba? Sa aning semester natin.
Usa ra gani, tagsa mo.
Ah, okay.
Oh,
Also,
ganahan kadugangan ba nagodot?
wala kay next week man daw. Wala man ka
Next week ang topic.
Ha.
Topic ba for approval?
Pero pagka last, masag,
Oh, last na. Syempre, last na. Dayon,
Sige,
pero
sige.
kuan, kung naatay time, ako na siyang ipapresent ninyo. Siguro, mopili ra ko og kisay mo present. So, kung ipapresent na ako, of course, you have to submit it before presentation. Kaya ako pa man ang basahon. Di ba?
sige.
Mm. So, inyo nang i-present. If napatay time, iampo nato na napatay time
Yan po na.
para ma-present ninyo. So that we will not discuss genetic engineering. So, ang inyong nga topic will be an application to any of the topics that we are or we will discuss in genetics. For example, you want to focus on taxonomy of come on, taxonomy of microorganisms using genotyping or using HTS, or you want to focus on certain studies or genetic engineering, dayon ang iyahang gigamit is CRISPR-Cas9. as its method. Ana. So, these are just some of the topics. So, sa genetics, man kung daghan kaayo kang pwede nga gamiton as your topic for review paper. Broad kay siya. So, kuan po ni kay daghan po kaayo ani sa kani. Ang sa genetics man gud is mura siya og pwede ka mo gamit og other disciplines. For example, sa systematics, ang systematics karon man gud, although naagi hapon they still use morphology as one of the characters in um in classification of organisms. Some tend to use genes already or DNA sequences in order to classify organisms, especially if we are talking about fungi and microorganisms. So we have discussed it in microbiology. Remember, sa microbiological aspect, Mango, we cannot just simply identify microbial species based on their morphology, right? Okay, especially sa iyang nga species, naapag na siya strain, and then the strain will also differ based on their physiology or their morphology. So there are a lot of characters that need to be considered. So most of the scientists or taxonomists right now use molecular markers for identification of or for the classification of organisms. Okay, so aside from of course morphological, physiological characters. So if you have observed in your systematics class, can you still remember APG4? Hello?
Sa mo.
Okay, sa APG4, kung makabantay mo, naka-download mo sa APG4, guys.
PG3
Naka-download.
ra ta mo nag.
Okay, basta na, nabot na siya APG4. So, imuhang lang taon sa APG4 is quite different from APG1 to APG3. Kay mo deal na guna siya dito mga ribosomal markers. Okay, mga molecular characters. So, trend na kaayo nga mogamit karon og genetic sequence for taxonomic classification. So, that's just one of the applications of genetics. Another kaning sa genetic engineering o sa biotechnology daghan kay kang pwedeng gamiton topic ani. In fact, there was this one paper wherein some of the genes sa kuan ni siya ha, sa agricultural aspect ni siya. So, ang ilahang gi-transfer is ang gene from an insect. Kay naaman tay crops, ang ilang ginabuhat sa agriculture man good is unsa nga mga genes ang ilang gamiton i-edit mo kuan na siya genetic mining. So, unsa nga mga genes ang gamiton nila in order to make a certain species of crop resistant to the insects or pests. So, what they do is they transfer the marker or the genes from a particular insect that can give rise to a character that is resistant to a pesticide or pest. Okay? So, ilang buhaton is to transfer that gene to a crop to make the crop more resistant to pests. So, naay mga studies inana. Okay? Anyway, kamo lang magpili sa inyong topics if unsa inyong ganahan for your semestral paper. So, ang deadline for topic approval will be next week Friday kaya? Or you want it extended?
Extended.
Okay. I want it extended as well kay para makapili mo tarong.
Yes.
Kay magpili-pili pa mo na ABCD. Siguro unsa man April 8 taron sa 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Wala ko'y kalendaryo so gaihap ko. 23? Sakto? Friday na April 23?
18, 19, 20, 21, 22, 23. Friday
Yes,
man.
ma'am.
Okay, so April 23 ang deadliest deadline. Charot.
20.
So 23 ang pero kung mupasa mo ugma dayon og topic, okay ra gihapon.
Sana.
Naga.
Ha?
Then RRL dayon, ma'am.
Ang kanang review paper Miss Abril is already RRL. Nandiyan ng RRL. Okay? So di na ko mo require o proposal for the laboratory. Kaya wala gyapon tay gamit. So murag instead ana murag siguro ari na lang ta sa review paper. Okay raman siguro na. Is it okay with you or do you want to make a proposal?
Review na ma'am.
Yan naman po may
No,
ano
review
eh.
ra, ma'am.
Review.
And of course, another requirement is the usual article critic.
Okay.
Okay? Always. Sige, article critic dayon na siya every topic. Sila naman ni 1, 2, 3, 4, 5, 6, 7, ano. Nanungha na di ay almost. Kompleto na ni?
Dili po.
Mura ba ni nang enroll? Mura ba ni nang
Ma'am.
enroll, no?
Naghan
Ma'am,
pa.
si
Kasi wala.
Gravador.
Ma'am, at least.
Si Gravador daw, ma'am. Wala
Napasal,
daw siya
ma'am.
kabalo if
Giovanni.
enroll pa siya.
Si
Then si
Gravador,
Rambay.
si Gravador wala na muna enroll si Gravador. Hmm. Si asa man is Montegreho?
Kapacheck up dahil si Rabay na kay
Naunsa man siya?
nabungol iyang left ear.
Naunsa?
Nabungol iyang dalunggan.
Na
Nabungol.
kuan. Nabungol siya.
Oh, yan.
Dalunggan.
Dalunggan. Sa iyang left.
Lisod pag mabungol ang mata, no?
Ang galing na to.
Diba? Lisod pag mabungol ang mata. Bito, kumusta si Montegreho man? Nga naman siya?
I don't know, sir. Basta, ano siya? Basta, kapacheck up
Okay.
naman siya.
Sige, fire.
Of course.
Okay, so ako i-share akong screen. Ay, saan? Okay. So, these are the books that we're going to use. Leyland Hartwell, you're already familiar with this. Diba, nag-assign ako to read Leyland Hartwell first chapter yesterday. So, I hope you have started reading the first chapter, Hartwell. It's entitled Genes to Genome, ang book. Okay. Coding kaayo. Basta, muna na siya. And then, we also have Hartel, Daniel Hartel. So, Daniel Hartel discusses linkage, gene linkage, and recombination quite detailed compared with the other books. As for analysis, from the name itself, maayo kayo ni siya sa genetic analysis ng book ni Griffith. So, katong na ay Mendel's law, the law of segregation of law of independent assortment, as well as extensions o mutation, ari na to kwaon sa Griffith. And then there are also some topics na ato na to kwaon sa Campbell. Depende sa topic. Hindi ka mo stick og isa ka book. Okay? Right. Maybe we can start with our discussion on sige, before that, unsa man ato i-fix na to 9 AM MW.
Night? That's it.
Lantawa sa inyong schedule if na by conflict babasig na i-conflict ko
Wala
mong
kayo, wala
sleep
for
no.
load
Okay,
sleep.
ang sa
Daan man yung load
ang
sleep.
sa amo ma'am. Sa ako ma'am kay sa Devi na mo kay 1 to 2 or 1 to 2 hapon mi dayon sa invert kay 5:30 beyond.
Okay, so ang kaning sa kuan siguro okay raman siguro. Atong schedule man gud is 9 to 10.
Sa
My worry is what if mo go beyond ko sa 10 ba? So, for example, 9 to 10:30 ana. So 30
Okay naman.
minutes.
Naay ko.
So murag ako ba feel na ko ako'y ga-enroll ninyo. I'm not sure
Kaya
na limot po ko.
Napoy lain mo.
Kay sa amo ang Devi pud ma'am kay 3 hours sa tato so 1 to 4 me. Pero
Okay
Tuesday ra.
ra. Okay ra kay hapon man. Ang akong concern is
Oo.
if taaba mo'y class of 10 to 11 MWF.
Na
Na
ethics.
mag-meet jud mo ana.
Ambot lang sad kay wala pa ako may
Murag magsabot naman pud daghan na. Sila.
Okay, so if ever mo conflict unsa man ha? What if TTH 10 to 11:30 in aron? Dili pwede mo?
Mo ka man tayo.
Imo na.
Ala oy gibalhin lagi og
Okay ra,
alauna akong time.
Ang gali chapter guys.
Wala lagi mo load sleep. Gibalhin og alauna akong time. So if ever walay conflict sa 10 to 11:30, naroon na lang ta 10 to 11:30.
Ang gal.
So
PPH na, ma'am.
dayon mo klase ra tag Friday if there will be presentation. Ako man gud especially kay solving ba, for example, maglami na mo og sulat-sulat din ha. So mo kuan yun ta mo go beyond good ta sa ato ang schedule. Mura na siya akong worry, what if ba mo go beyond ka sa schedule and then ay mo conflict sa inyong schedule, naka-absent mo. Okay. Pero anyway, if na ay conflict, feel free to inform me via chat na lang if ever there will be conflict in schedule. So siguro stick
Yes,
lang
ma'am.
usap ta sa 10 to 11:30 every PPH. Okay ra?
Okay na, ma'am.
Okay na, ma'am.
Okay, sige. O change sa dayon ta if na ay conflict.
Ikaw na general lang mo.
Oh, pero wala ko katimaan unsa tong mga rasa pa akong gi-enroll. Basta wala mo'y conflict. Wala mo'y conflict sa ako na inumduman.
Hasta sa minor, ikaw may dapat.
Oh, ako tanandong.
Na-enroll may advice
Pero ang Invisay ninyo nalimot ko giusa ra ta mo sa Invisay. Nalimot lang po ko unsa po tong
Appeal me, ma'am. Third
Kamoy
year.
labo third year. Ang sa third year ang akong
Okay, wala.
enroll is GE11 and 12. Pero
Ay,
kanang
oh,
G11
okay.
and
Di lang
12
na-
ako na gani nang gi-overload, gi-justify na nako didto. So dili na gid ta mo pwede add og GE10. Overload na gud gani na. Gi-justify na
Yeah.
nako didto. So di na ko ka-add og GE10. So maybe you can enroll GE10 and Philippine Lit. Usog man gud og kuaning CHED go di po ta
Sayang
pwede
ka sa
mo
mga
dili.
ulit, ma'am.
Nya di po ta pwede kabuot sa CHED maingon og ayaw na lang to gi-enroll. Masangit man gyapon mo apply mo for graduation po. Maybe you can have it enrolled siguro first sem para
Kasi
wa na
na kayo.
mabilin. Yes. Okay.
Excuse me, ma'am.
So, yes.
What about sa biophysics, ma'am?
I'm not sure with biophysics kinsay pandulo no man pud sila giingon.
Kaya ni
Biophysics.
silang to po ko sa mga kuan pages sa katong geo og physics kay wala pa man po post.
In fact, among load karon except sa biology subjects are still subject to changes basig napagani na reshuffling. Shuffling pa mi ani. Except for biology subjects. Ang biology subjects ano na siya, fixed na. So ako na jud din yung genetics.
Okay.
Dayon, pero kanang sa minors, ang boto pa na klaro. So badunon pa na didto kay ang physics and geo may nag-arrange ano. So basig kanang inyong hapong section wa pa pud siguro na natarong og kinsay mo handle ana. So, ulat-ulat lang.
Sige ra pag
Yung
naulahi.
inyong load slip ni saan na mo request sa registrar email?
Mana, ma'am? Ato pang enrollment.
Okay, sige. Ulat lang basig busy pud sila. Okay,
Mm.
sige. Let's start with our discussion on the first chapter. This is found from the book Hartwell et al. I'm not sure. Wala pa ko ka-visit sa DriveGen if na ay new edition ani. Last time mo pa man ni latest ng 4th edition. Let me check later on. Nag-download mo lain nga book or katura po ako gi-post.
Katara may post.
Okay. So, maybe I will check on nga if naabay bagong edition ani ka Hartwell. O man po ko ka-check. If naay bagong edition ani katula tong ganito. Okay. So, genetics is defined as the study of biologically inherited traits. And then there are also major advancements in the development of genomics. So what is genomics, by the way?
But,
Hello? What is genomics?
study of genomes.
Guys,
Study of genomes.
nahilom na. Ha?
Study of genomes.
Study of genomes.
Study
Okay. And then it only started 3 years after Mendel reported his experiments on Pisum sativum. So he spent a lot of his time before in studying inheritance in T. sensitivum using different morphological traits. So because of his extensive study on how uh, morphological traits are being inherited by the offspring or progeners, and then how is it or how some of the traits are independently assorted. And then there were also um, several mix of different traits from one generation to the next in his experiment. He has actually published or he has reported his findings about the law of independent assortment and law of segregation. Actually, Gregor Mendel was quite lucky at that time. Okay, if we are to discuss extensions to Mendel's laws, there are a lot of factors that need to be considered other than law of independent assortment and law of segregation. Okay. At that time, he was quite lucky kay very distinct man ang separation of morphological traits. But right now, nanatay ginatawag og environmental effect or environmental factors that affects gene expression. Okay? So at that time, very distinct ang pag-separate sa traits atong gistudihan ni Mendel. So walay problem sa iyahang results. That's why smooth ra tong iyahang sa law of segregation and law of independent assortment. However, after several years, Friedrich Miser discovered a new type of weak acid abundant in the nuclei of white blood cells, and this weak acid is known as DNA. So it was also discovered that DNA carries the genetic information of a particular species. Okay, so DNA, as you all know, is the first macromolecule that is made of nucleic acids. So it also serves as the repository of the genetic code. So these genetic codes are directed by sequences of uh, nitrogenous bases. So we have four nitrogenous bases. So I suppose or I assume you know already what's the difference between a nitrogenous base, nucleotide, nucleic acid. Okay? So kabalo na mo sa different parts of what is the structural unit for nucleic acid, particularly DNA molecule. So, uy,
Kasi yun.
Yes, ma'am.
what's the structural unit for DNA molecule? Building block.
Deoxyribosugar sugar phosphate.
Building block.
Yes, nucleotide.
Okay, nucleotide. And nucleotide is in turn composed of, yes, Miss
Nucleic
Elmas,
acid.
what is it?
Deoxyribosugar sugar phosphate
And,
and nitrogenous base.
okay. So, if we are to compare DNA and RNA, first, DNA is double-stranded, RNA single-stranded. DNA contains four nitrogenous bases, that's A, T, C, G. RNA, T is replaced with U. Okay. So, if we are to compare the structural composition or the structure of thymine
Ano,
and
Christmas?
uracil, na siya slight difference. And then, in terms of functional, they also differ. So DNA carries the genetic information. At the same time, it directs production of proteins that are necessary to maintain life. Okay. DNA, aside from being the repository of genetic information, they are also considered as the molecule of heredity. And remember, in basic biology, one of the property or manifestations of life is to have or possess genetic program. So naagid tanan organisms are considered or are considered to be alive should have a or should possess a genetic program stored in the DNA. Now DNA is very important, or the diversity of DNA generates a lot of living organisms, species of living organisms that we can see today. So here we can see bacteria, and then we also have dolphin, plants, mouse, and humans. The difference in the biological or in the biological information in DNA is responsible for the enormous diversity of living organisms. But despite being different in their DNA sequence, there is a common ground which is responsible for or what do you think is common for all organisms in their DNA makeup?
Kaya development nila, ma'am.
Are you sure about the development? Remember, organisms assume different modes of development, in the way of development. But if we are to relate it in the DNA, what do you think is common to all organisms from the tiniest living organisms to the largest living organisms? What is common to their DNA molecule? How did we come up with that last universal common ancestor theory or concept? Luca, for example, di ba na sila giingon dito last universal common ancestor, or all organisms that are living today, or all species of living organisms, came from a single ancestor. What do you think is common at the molecular level? Yes, please.
The same genetic code, the four letters.
Yes. Okay. All organisms use the same genetic codes. That's ATCG. And in terms of the molecular structure of the DNA, all organisms use the same DNA structure. Okay. The only difference is that we have different genetic or DNA sequences. We differ in the sequence, in terms of the sequence, and at the same time in terms of gene expression. Okay. Naamantay ginatawag og gene expression. It may be less expressed, overexpressed, and then there is also consideration of when and how these genes are expressed. Natay ginatawag og mga regulatory networks for gene expression. Now, biological information is encoded in the nucleotide sequence of DNA, and each unit of information is discrete. Okay, so there are some techniques now that uses computers in order to handle DNA sequences. So, mention ako ganina one example katong sa DNA microarray chips, and then there is also HDS, high throughput sequencing, NGS, next generation sequencing, and many others. So daghan ni siya nga mga techniques. Lain-lain po ni siya og mga equipment nga gigamit in order to identify DNA sequence. Now the problem is after identification of DNA sequence, how is it used? Diba? So there are also some genetic techniques in which they identify the genes that are expressed in a particular time. Okay? So napoy mga techniques nga mogamit o or there are some techniques that uses machines in order to identify which genes express on what time. Okay? This is very important in order to know the function of each gene. Okay? Para kabalo sila unsay function sa gene. And if we are to relate it in the medical aspect or medical context, they need to know which gene is expressed in order to produce or cause a certain disease, or which gene is expressed in order to counter that particular disease. Okay? So aside from identification of the DNA sequence, we also need to identify which genes are expressed and how they are expressed. Before we continue, it's important that you know the difference between genes, DNA, chromosome, and genome. Okay, can you define these terms? how they are different. Let's start first with genome. What is a genome, Mr. Solera? Hello? Kadungog ra mo nako or naputol na po ko? Can you hear me?
Yes, ma'am.
Hello? Okay, sige. Mr. Solera, what is a genome?
So, a genome is an organism's complete set of genetic instructions.
An organism?
Organism's complete set of genetic instructions.
Okay. So do you mean we have to add all the genetic material that is found in the organism regardless of their number of cells in order to identify the genome?
No, no, ma'am, na naman siya kanang kuan. Set of genetic instructions encoded within an organism cell. So each instruction naman siya, di ba, specific function.
No. I'm talking about genome.
Mm.
Okay, do you agree with the answer of Mr. Solera, Miss Abril?
No, ma'am.
So what is a genome?
Genome is a complete set of genetic information in an organism.
Imorang gi-repeat ang answer ni Solera. How about you, Miss Tubel?
Each I think each genome composed of information like contains all the information needed in the in an organism.
Okay, who wants to answer my question? Ay, ginoo ko. Who wants to answer my question? Do you have any idea, Mr. Gador?
Kasi question niyo mo.
Um, maor maora to murag complete siya nga ano siya?
Okay, Mr. Banagudos, how about you?
Agree ako. Agree ako. Complete.
The same answer, Elnas?
Ang genomes, ma'am.
Hmm.
Ang genomes are kanang the entire collection
The library.
of chromosomes in each cell of an organism.
Okay. It's important that we also include
Chromosome.
cells. Okay. But there's one thing that's quite maybe something that we have to correct. It's not a chromosome because not all organisms contains chromosome.
Okay. Sa human,
Yeah.
ma'am, in the case of
In the case of human, yes, that's correct. Entire genetic. So to make it safer, and instead of using chromosome, maybe you can use genetic material, complete set of genetic material in each cell instead of organism. Because if you say in each organism, then you have to calculate all the genetic material for a multicellular organism, which is wrong, right? And not all organisms are composed of only one cell or unicellular. Some are multicellular. So in order to incorporate or in order to make it a bit correct and ma-appeal po ang multicellular organisms, we can use the entire set of genetic material that is found inside the cell or inside the cell for a particular organism. Okay? How about DNA molecule? Oh no, not yet. Genome, then
Chromosome.
chromosome. Yes. Who wants to answer? What is a chromosome?
Huh?
Volunteer? Okay, let's start again with Mr. Solera, then Miss Abril, Tubil, and then followed by kasi pasunod ang inyong names. Una si Solera, Abril, Tubil, then followed by Gador, then Banagudos. Elnas, then Triplet. Okay. Sige. Let's start with you, Mr. Solera. What is a chromosome?
So a chromosome are thread-like structures located inside the nucleus of an animal and plant cells. And they are made up of protein and a single molecule of DNA.
Animal,
Yes.
animal and plants only.
Ay, other, also po, ma'am. Tanan.
Okay,
Pero,
how are
there,
chromosomes? Sige, go on.
wala na, ma'am. Okay na po.
How are chromosomes different from DNA, Miss Elmas?
Chromosomes are different with DNA in the sense that a DNA molecule is assembled into a long strand of chromosome in the case of plants and animals.
Mm-hmm. How about you, Miss Triplet?
Ma'am?
How are chromosomes different from DNA?
Yes, ma'am. Chromosomes have they certain proteins called histone proteins, and most eukaryotic organisms have 8 histone proteins, and for prokaryotic, 4 histone proteins.
Okay, that's correct. DNA molecule is a long thread of thin-like um, no, DNA is a long thread of long thread molecule, which is packed together by proteins called histone proteins, and that's correct. One of the difference between prokaryotes and eukaryotes is the number of proteins. So once this thread-like structure, the DNA molecule, is um, packed by histone proteins, they're usually called as what? The term for that,
Sama?
bead-like structures. The bead-like structures, kind of
Nucleosome.
ganing, yes, that's correct, nucleosome. So once this DNA molecule is packed by histone protein, it forms a bead-like structure called as nucleosome. Okay. 8 for eukaryotes, 4 for prokaryotes. And then the main reason here is to um, um, prevent DNA molecules because DNA molecule is quite long, and then to prevent them from entanglement, they are packed together by histone proteins and form chromosomes. Four, of course, actively dividing cells, and chromatin for non-dividing cells. Remember, chromatin starts to condense during cell division. At what particular stage or step of cell division, if you can still remember, when
Huh?
does it start to condense? Huh?
Two.
Huh? When does chromatin start to condense and form chromosomes?
Inter, in
Cell
the
division.
G2 phase,
Be specific.
interface in the G2 phase. Oh, ba?
Really?
Tayo, tayo.
Hmm? Who wants to answer my question? At what specific stage or step of cell division or cell cycle? This is just a review of basic biology. I think this was well discussed in botany and zoology before. I assume you know this already. Na-discuss mo po ninyo yung bio tutorial, right?
Wala.
Okay. Silent.
Prophase.
Okay? That's prophase. Okay, last term, genes, before we move on. What are genes? And how are they different from DNA? Let's go to Miss Tubil.
Genes are made up of DNA, while DNA is long chain made up of nucleic uh, like adenine, yeah, ATCG, mo to, ma'am? Like genes are made up of DNA, and DNA are long chain that are made up of ATCG and A.
Okay, do you agree with the answer of Tubil, Mr. Banadudos?
Oh.
Hello? Managudos. Managudos, are you
Hello,
still with
hello, hello.
us?
Wala, gikan kang ni. Asa?
Okay, do you agree with the answer of Tubil?
Asa
Oh,
answer ni?
nangihiday ka. So, kindly repeat your answer, Miss Tubil. Kay para na madunggan ni Banagudos. Kay nangihis Banagudos. Tubil. Tubil, Tubil.
Yes, ma'am. Naka-off mic tayo. Sorry. So,
Okay.
genes are made up of DNA or RNA, while DNA is a long chain of polynucleotides such as ATC DNA.
Okay, do you agree? Nadungog na ni mo Banagudos?
Yes. Agree.
Okay, so, ha?
Agree.
Agree ka?
Oh.
How about you, Gador? So, guys, Banagudos, eh?
So muda ang DNA to
Agree.
ang
Uy.
so mo ko sa genes. So ang genes, ma'am, is the region of the DNA in which it encodes
No,
or
sakto.
it encodes the protein.
Okay, another definition. Who wants to volunteer? Aside from those mentioned by your classmates, another definition of a gene. And how is
Ah.
it different from DNA? Any other answer from the group? Abril?
Ma'am,
Hmm.
ang gene naa siya sa kuan usa ka portion sa DNA which has composed of nucleotides which includes protein.
Okay, the same answer with Gador. Any other answer? Miss Kaplit?
Yes, ma'am. In DNA, there are also segments or part that are non-coding DNA, and those non-coding DNA do not code for specific proteins, which is different from genes. And this non-coding DNA also have their function. Some say it's regulatory function or structural function, and also they also contribute
Senador.
to the diversification
Senador.
of genes.
Okay, so that's correct. Any other answers? Any other answer aside from those?
Mag-maura
Do
dyan
you
na.
have any additional information, Miss Elnas? Hello? Hello?
Yes, ma'am.
Yes.
Isa kayo na ini ko sa
Hmm. Any other answer? Any other definition of a gene?
The genes, ma'am, are kanang unit of heredity. Kaya it can be transfer the characteristics from a gene can be transferred
How is it
from
different
parents
from
to children.
DNA molecule?
As what Margav said, ma'am, the difference are those katong intron o extrons ba tong part nga dili siya magamit for encoding for proteins.
Okay. As a follow-up question, since you mentioned introns and exons, are introns always classified as introns and are exons always classified as exons?
Ha? Ha?
Hmm? Are introns always introns and exons always exons?
No.
Okay. When are they
Um,
classified otherwise? Yes?
in alternative splicing, ma'am,
Okay,
ang
that's correct.
introns contains.
Okay, thank you. At least you can still remember our discussion last time in um, protein synthesis. I hope everyone can still remember alternative splicing. So, genes are actually components of the DNA molecule. So, Ms. Tubil, correction to your answer. You said DNA is component of genes. It's actually wrong
Ha?
because genes are component of the DNA molecule. So a DNA molecule
Nabaliktad eh.
contains hundreds of thousands of genes depending on um, the size of the DNA molecule. Okay, so answer of split is also correct, wherein these genes can either work as a code for certain proteins, and they may also serve as regulatory networks. And the name itself, it regulates. Okay, what does it regulate? Its expression, gene expression. So these regulatory networks are usually, okay, not all the time, are usually found adjacent to the gene that codes for a protein. For example, okay, gene A, which is adjacent to gene B codes for a protein. Gene A can regulate expression of gene B. So, pwede niyang i-overexpress, pwede niyang i-express moderately, or pwede niyang i-suppress. Okay. So, those are the function of regulatory networks.
Excuse me, ma'am.
Yes? Di madungog?
Oh, okay na day, ma'am.
Oh, okay na? Nadungog ako ganina?
Dili ang slide mo.
Ang slide man di nakita.
Okay. Nakita na ninyo ang slide.
Yes,
Dapat
ma'am.
mag-move na ko. Okay. Sige, go. Pero maklaro ra kung tingo guys, pag sulti mo og kuanhan, putol-putol ko kay kaning globe. Kay kaning globe mo kalitra ni siya to Eric.
Mauro po nag-slide.
Okay. Basta pag sulti ra mo dili na maklaro. Feel free to interrupt. So here, it's genes are identified or described as sequences of DNA that encode proteins. So in the human genome, there are 24 kinds of chromosomes. Okay, and there are or it encodes 20,000 to 30,000 genes. So, chromosomes are described or defined as organelles that package and manage the storage, duplication, expression, and evolution of DNA. In terms of their structural makeup, they are composed of DNA plus histone proteins. And genomes are the entire collection of chromosomes in each cell of an organism. Okay. So I always emphasize the cell part because remember, organisms can be multicellular or unicellular. Why is it 24 kinds of chromosomes? Remember, there are only 23 pairs in the case of human genome. Why is it 24 kinds? Any idea? Kasi wala pa natawag. Yes, Triplet, you want to answer?
Apil na ang sex chromosomes, ma'am, X and Y.
Yes. Why is it 24 kinds? Okay. Can you elaborate your answer, Mr. Flip? You're actually correct, but I just want it to be elaborated.
Katunan na yung mga special child lagay, ma'am.
Kay sa Abril. Masyaktaan ng special child, Abril.
Ang 22 autosomal chromosomes, ma'am. Then, ang
And
other
then?
2 are sex-determining X and Y chromosomes.
Okay, that's correct. So 22 are autosomal chromosomes, which means they are homologous chromosomes. So one maternal, the other is paternal. That's why there are 22 kinds. Yeah, 24 siya because the other 2 are sex chromosomes. So remember, X and Y chromosome are different from each other. That's why there are 24 kinds. Okay, is that clear?
Yes, ma'am.
Okay. Very good. Alright, can you still remember our discussion in microbiology about Griffith's demonstration of bacterial transformation? Can you explain how did Griffith's demonstration of bacterial transformation prove that DNA is the genetic material? So I know you know what is bacterial transformation already, but what I want you to explain is how did it implicate that DNA is the genetic material and not any other um, molecule inside a cell. Who wants to explain? Volunteer? Volunteer. Uy, people of the Philippines. Hello?
Ato.
Hello? Nawag na po ko.
Ayaw, ma'am.
Hari kong Gador. Gador, please answer the question.
Wait, sir.
Sige. I'll wait for 5 minutes. Okay. So, anyone can answer after 5 minutes if you wish to answer. I'll be back after 5 minutes. Okay na? Wala pa 5 minutes. Kindly relate Griffith's demonstration of bacterial transformation to how he proved that DNA is the genetic material.
Ito ng mouse. Ma'am. Ito ba?
Kato ng mouse, dura o. Griffith's experiment. Yes. So, I know you're familiar with this, di ba? Na-discuss man is microbiology, bacterial transformation. I-discuss mo nila ako ng Griffith's experiment, right?
Agway.
Na-discuss
Yes,
nila ako,
ma'am.
di ba?
Yes, ma'am.
So, na-discuss is microbiology. So, I suppose you know what is Griffith's experiment all about. So, instead of explaining what is Griffith's experiment all about, can you relate its result to how it proved that DNA is really the genetic material? Who wants to explain? Do you want to answer the question, Miss Replit? How about you? Nahuman naman is Gador, no? Banagodos,
Ito isa.
Silent Night Pod. Si Tubil. Do you want to answer the question, Miss Tubil? Elnas? Soliera, Abril? Who wants to answer the question? Kindly answer the question. Relate how Griffith's experiment proved that DNA is the genetic material. Okay, simply because during transformation, di ba? What happens during transformation? Kanina lang. What happens during transformation? Bacterial transformation, ha? Not data transformation. Bacterial transformation. Wala gyud po yung answer? What is bacterial transformation? Uy, nahuman, namutanag microbiology, pag sure. Mr. Banagodos.
Kuan. Gene transfer sa naked DNA sa environment.
Okay, is it gene transfer or genetic material?
Genetic material.
Okay, so genetic material transfers from the environment or is incorporated by the cell from the environment. So there is transfer of genetic material. Okay. So actually, Griffith's experiment proved that DNA is the genetic material because after transformation, there is also a corresponding change in the morphology of the bacteria. Okay. So right after transformation, the morphology of the bacterial cell also changes from which is virulent. Is it the smooth or the rough
Smooth, ma'am.
bacteria? Okay, it is.
Smooth.
Okay, it's the smooth,
Smooth
which
strain.
is pathogenic. Okay. Now, one another important concept of DNA is the double helix model by Watson and Crick. So here it shows the different nitrogenous bases. We have ATCG, wherein A and G are larger and composed of two rings. They are also classified as purines. Okay, and T and C, cytosine and thymine, are relatively smaller compared to purines. They are only composed of one ring. And then these nitrogenous bases is one of the components of nucleotides which serve as the building block for DNA. Okay, so together with phosphate group and together with the phosphate group and deoxyribose, they form a nucleotide which serves as the building block of the DNA molecule. It's called it as a nucleotide, and then they compose the DNA molecule. These nitrogenous bases are connected to each other, or there is a bond present between these nitrogenous bases. We call this as hydrogen bond. Okay, so DNA consists of two long chains of subunits twisted around one another to form a double helix, which is in contrast to RNA composed only of one strand. Okay, and then the subunit of each strand are nucleotides in which they differ on their nitrogenous base. Okay, so nucleotides differ based on the nitrogenous base present in that particular nucleotide. So all nucleotides contain phosphate group and deoxyribose, and they only differ structurally on their nitrogenous base component. So according to Erwin Chargaff, or also known as Chargaff's rule, the number of adenine is approximately the same with thymine, which is the reason why they concluded that adenine and thymine are paired, while cytosine and guanine are said also to be complementary. So according to Chargaff, if they are to pair adenine with guanine, the size of the DNA is too wide, and that's because both adenine and guanine are large molecules. They are composed of purines, whereas if it's pyrimidine to pyrimidine, then the DNA is too narrow. Okay. So the base pairing between A and no, sorry, the oppositely oriented strands are said to be antiparallel. So one strand starts from 3 prime to 5 prime, and the other strand starts from 5 prime to 3 prime. Okay. Now, proteins are also considered as polymers of hundreds of thousands of amino acids, depending on how big or what is the size of a polypeptide or a protein molecule. By the way, how are proteins different from polypeptide chain? Before we continue, how are polypeptide chain different from protein molecule? Abril? Abril.
Yes, ma'am.
Hmm. How are polypeptide chain different from protein molecule?
Polypeptide chain is composed of different protein molecules.
Are you sure?
Amino acids.
How about you, Soliera? Um, Maragodos?
Kuan, ang polypeptide chain kay chain of amino acids sa siya, while protein are long chains of amino acids. So mas contain of one or more polypeptides ang protein kaysa sa polypeptide chain.
Okay, that's correct. A protein molecule may contain one or more polypeptide chain. Okay. So in terms of the size, polypeptide chain may be smaller compared with protein molecule, and polypeptide chain, protein molecule may be composed of one or more polypeptide chains linked together by different chemical bonds. Okay. So,
Peptide.
information in DNA of a gene dictates the sequence of amino acids for the protein. So, the sequence of nucleotides in a particular gene determines the type of amino acid, no, the type of protein or polypeptide chain produced at the end of protein synthesis. Okay? So, this order of amino acids determines the type of protein and its three-dimensional structure. Okay. So aside from the order of amino acids, what else determines the three-dimensional structure of a protein? If you can still remember our discussion in botany before, what are the different characteristics of a polypeptide chain, or what determines the three-dimensional structure of a polypeptide chain at the end of protein synthesis? One is the order of amino acid. What else? Can you still remember? Aside from the order of amino acid. Yes? Yes? What's the answer? Kinsa to ini on sa mic? I think it was triplet or Elnas or Abriel.
Mo-form na siya usable, ma'am. Mukha na siya protein. Three-dimensional.
Unsa kay
What
question no?
determ what determines the three-dimensional
Bonds.
structure of protein? Yes, Elnas?
Iyahang bonds, ma'am.
Bonds?
Peptide bonds, charot.
Iyang kuan,
Peptide bonds? Are you sure?
iyang number of amino acids.
Okay. Number 2, number of amino acids. So first order of amino acids, number of amino acids. What else?
Type.
Okay, type of amino acid. And lastly,
Napa di. So,
yes, napa, last.
sequence.
Sequence.
Okay, sequence of amino
Ah,
acids. Order, number, type, sequence.
Order
Okay,
number types.
yes, that's the three-dimensional structure. Three-dimensional structure refers to what structure? Protein. Is it primary, secondary, tertiary, quaternary? It's for you
Tertiary.
to figure out.
Sa to,
That was
ma'am?
discussed in botany. Okay, So na siya three-dimensional structure.
There siya.
Okay. So a protein may contain two or more polypeptide chains. That's why it assumes a quaternary structure. That's quaternary structure. A very good example is what?
Ha?
Hemoglobin. Okay.
Hemoglobin, globule proteins.
Okay. And then diversity of three-dimensional structured protein generates an extraordinary diversity of protein function. That's why um, different genes can produce a wide array of proteins. And then proteins can also be very specific. Okay. And then plus alternative splicing on top of that, it can produce a wide variety of proteins. And proteins um, does not produce only one type of, I mean, proteins do not only function one specific role in a certain metabolic activity. So there are some proteins that may function several important or key roles during chemical reactions, okay, in the organism. The chemical formulas for two amino acids. So these are just two out of the 20 types of amino acids. We have alanine and tyrosines made up of the amino group, central carbon atom, and then we also have the carboxyl group. Carboxyl group there is colored blue, amino group is colored green, and then we also have C, central carbon atom, and then connected to the R group, which is colored orange. So orange. So what determines the type of amino acid is the structure of the R group. Okay. So these proteins assume the three-dimensional shape because of the particular chemical bonds formed between amino acids. So if an amino acid which contains a sulfur binds with another amino acid that contains sulfur, it will form a disulfide bridge. So for the secondary structure of the protein, we have alpha helix and then we also have beta pleated shape. So here we have hemoglobin beta chain. So this hemoglobin beta chain is just a component of the hemoglobin molecule. And of course, we also have lactate dehydrogenase. It's one of um, example. So RNA may have been the first information processing molecules. If we are to relate this in evolution, it was believed that it was RNA that first carried the information, genetic information. But RNA is a very unstable molecule. That's why it binds with another RNA molecule forming a double helix structure, which we now recognize as DNA. So before, it was RNA that carries genetic information. In evolution, we call that as RNA world because what is predominant at that time was RNA molecule instead of DNA. Then after several years, DNA took over the linear information and replication functions, and RNA now serves as an intermediate molecule during protein synthesis. Okay. Although RNA is very unstable, it still plays an important role during protein synthesis. Okay. So proteins took over the three-dimensional folding functions, and all organisms alive now descended from the first organisms that adopted this molecular specialization. This is also one of the reasons why we believe that all organisms came from a common ancestor because of the molecular makeup. Okay, we share the same molecular makeup, and some of these molecules assume the same function for all organisms. Example, all organisms undergo protein synthesis in order to produce proteins. Okay? And they use their DNA as a template for the production of RNA, which serves as an intermediate um, substance or intermediate molecule in order uh, to be translated to form proteins or polypeptide chain at the end of the process. Okay, so here is an example of how RNA evolved as an intermediate molecule from in order to convert DNA information into protein. Okay, so according to the central dogma of molecular genetics, DNA serves as a template strand that is converted or transcribed into an mRNA molecule, and mRNA molecule is translated into polypeptide chain or protein. As an example. So, after protein synthesis, does not mean that the product is already a protein molecule. So, the product is actually a polypeptide chain. And this polypeptide chain, according to cell and molecular biology, still undergoes protein folding. So, that is another separate process in order to generate the diverse protein molecule. So right after protein synthesis, napa dyan na siya other process, we call that as protein folding. So mag-assume na siya of different shapes, which is, of course, needed by the cell. Okay? So you're familiar with this already, the different codons and the corresponding amino acid, or the amino acid that they code. So an amino acid may be coded by several codons, but a codon cannot code for several amino acids. So genes code for proteins. Proteins control the chemical and physical processes of cells known as metabolism. That's why proteins are very important. Okay, and then some of these proteins act as enzymes. Remember, proteins are classified into 8 types based on their physiology or physiological function. Okay, so Arkada Bogorod introduced the term inborn errors of metabolism, which can be caused by abnormality on the protein that is produced by a particular diseased organism. Okay, so an example for that is alkaptonuria, which is also known as black urine disease, caused by abnormal substances present in the urine of an infected or diseased individual. Okay. Which do you think is the main culprit why there is production of abnormal substances in the urine?
Mutation.
So among these molecules, which do you think is responsible for the production of um, abnormal substances in the urine? Okay. DNA might be the reason. So if, for example, there is mutation in the DNA molecule, it will produce a different RNA molecule depending po sa iyahang site for um, mutation. It could be point mutation, or there could be a mutation in a segment of the DNA. So that can cause a great effect on uh, the polypeptide chain that will reproduce at the end of the process, or maybe it's the enzymes that are non-functional. So if the enzymes are non-functional, or there is error during transcription, so those are some of the factors, or might be the reason while there is production of abnormal substances. Okay, so basically the this is the normal pathway of how uh, normal individuals produce substances in their urine. So the more normal pathway, there is presence of phenylalanine and then enzyme 1, okay, production of tyrosine, and then another enzyme, so on and so forth. So which enzyme causes the production of, or which enzyme causes abnormality?
Kanang HA oxidase.
What is responsible for the abnormality in alkaptonuria? What enzyme? Based on this diagram. Yes, triplet.
It's the absence of the enzyme HA oxidase, which
Okay.
in turn accumulates.
Okay. So HA oxidase is non-functional. Therefore, this homogenesic acid or HA accumulates. What do you think is the function of HA oxidase? What do you think is the function of HA oxidase?
Oxidizes the homogenistic acid.
Oxidizes, okay? And so, after it oxidizes homogeneous acid, what happens? It is
It
transformed or it is converted into,
allyl-acetoacetic acid.
yes, that's correct, until different enzymes are also responsible for the conversion of this acid into carbon monoxide plus water. However, due to a non-functional HA oxidase, HA or this acid starts to accumulate and turns the urine black in air. That is due to um, um, the stop in the pathway. Okay. So aside from the role of the DNA as a template for production of proteins, there is also evidence that the difference in the DNA sequence is responsible for the divergence and the diversity of the organisms that are now present. So fossil evidence that were discovered previous years ago showed that there is really stages in the evolution of life. Okay, so if you can still remember in our discussion in microbiology, there is presence of stromatolites before, and then there are also some prokaryotes that were preserved in rocks and discovered and are believed to be present 3.7 billion years ago, and the first single cell eukaryotes to the formation of early multicellular eukaryotes and Cambrian explosion. So this ano, um, geologic timetable will be discussed in evolution later on. Okay, so it shows that divergence and mutation and different mutation that occurs within a particular gene family causes a diversification of ancient or primitive organisms. So this is an example of gene duplication in which, for example, we have here ancestral gene A undergoes duplication and then it forms two exact copies of gene A. However, further duplication causes a divergence in this gene A family. So, if for example, this gene A family or the gene A undergoes mutation and DNA rearrangements, it forms different genes that are somewhat related to ancestral gene A. So further duplication and divergence from for mutations and DNA rearrangements. So this gene A with a superscript 1, 2, 3, and 4 were actually um, belongs to a family of gene which belongs to ancestral gene A. So, some of these genes are obvious in the genetic makeup of some organisms. So these changes in the genetic makeup can cause diversification at the same time, changes also in their physiological function. And of course, the proteins produced at the end of protein synthesis. Okay? Another thing, there might be, aside from the change in the polypeptide chain that it produces, there might be also change in the key regulatory network. So if there is a change in the key regulatory network, there will also be a change in on how these genes are expressed. Okay, remember, it's the regulatory network that moderates or that controls expression of the genes. So example, here we have two-winged flies evolved from four-winged flies. So instead of producing four wings in this particular fly, um, the regulatory network starts to um, less express that particular gene and produce hoters which are responsible for the balancing of flies when they are flying or when they fly, okay, or during flight of these flies. So mutation of a regulatory network converts a normal two-winged fly into a four-winged fly. So if so what is responsible for the overexpression of these wings is the regulatory network, mutation of the regulatory network. Okay. So it doesn't mean that it's always the gene that codes for the protein that is mutated or diverged from the original one or from the ancestral one. It could also be the regulatory network that directs the expression of a particular gene. Okay. So here we have different modern genetic techniques which will be reported by who wants to volunteer.
Wala.
Okay, who wants to volunteer? Maybe we can start with unsa alphabetical ta?
Hindi ako.
Hmm? Or
Yes, ma'am.
mag-random na pug ko using computer. Sige, mag-random na lang ko using computer. And then I'll just send you your topics. Short reports ha. Remember kato kung giingon. Importance, you have to emphasize on the importance of the genetic technique, its advantage, disadvantage, and related studies that uses that particular genetic technique. Okay? So short reports lang is siya. So there will be four topics. We have genotyping, and then we also have HDS, NGS, 5DI. So first genotyping, HDS or high throughput sequencing. We also have next generation sequencing, and then also have the fourth one is DNA. This one is actually found in Hartwell, Nisha, DNA chip, and then lastly CRISPR-Cas9. Okay, so there are five topics, and then and the sixth reporter, turot good demo. Ang sixth reporter will present the article. So, kani siya may pinaka-last ang article. We'll just post the article later sa Google Classroom. Okay? Question?
Gosh.
Question before we end this call. It's almost 12.
Kano sa ka
Siguro
mo wait sunod mo.
Tuesday next week, same time, 10. Pero if naamoy conflict by the time nga madawat ninyo inyong load sleep and then naay conflict sa ako ang dili nga schedule, please inform me so we can adjust. Ha?
Sige.
Okay.
Ma'am,
Hmm.
speaking sa imuhang kuan, giingon nga new edition sa katong ka Hartwell kay naay 6th edition.
Okay, sige. Kana lang ito i-download.
Na.
Kabalo
Na-download
naman
na
mo
ako,
mo
ma'am.
down. Okay. Kung kabalo mo download, please download that file and then we'll use that as our main reference. Okay?
Ako na lang i-send sa Gmail.
Okay, it's up to you. You can also send it. I think dili siya pwede sa Gmail kay ang maximum sa Gmail is only 25 MB. So maybe you can
G-Drive, ma'am,
upload
then.
it in Google Drive and then send us the link. Please add Mr.
Yes, ma'am.
Soliera and Miss Abril sa ato ang group chat, Mr. Gador. And then remove your classmates nga wala na enroll sa genetics.
Yes, ma'am.
Thank you. Wala na question or concern?